Thanks for joining me on the second video in our series of lecture material on a chapter about blood. During this video, I aim to focus on reviewing the composition of blood and then moving to study the red blood cells in a little more detail. On that premise, let's get going with the material. So to start off this section, I want to say a few words about what blood is made of. Interestingly, blood is the only fluid tissue in the body, and we remember it was classified as a special type of connective tissue. So let's have a closer look at what blood is made of. The liquid matrix forms about 55% of the volume of blood. This non-living fluid is called plasma. All other components of blood, and by this I mean these cells, are suspended in it. So let's have a look at those. These cells are known as formed elements of the blood, and there are three types that I want us to talk about. Firstly, about 45% of the volume is red blood cells. These are known as erythrocytes, and often you will notice that I abbreviate them simply as RBC, short for red blood cell. We also have white blood cells, which are abbreviated as WBC. The proper term to describe these is leukocytes. And finally, the last group of blood cells that I wanted to discuss about is platelets. These play an important role in clotting of the blood. So we have about, or actually less than, 1% of each for both white blood cells and platelets. And I should probably say that there is a little bit of variation in these percentages of the volume for these components of the blood. As a quick review, let's go through the percentages of the total volume again. The number of red blood cells, which we knew as erythrocytes, was about 45% of the whole blood volume. This is known as our hematocrit, and it varies a little bit between males and females, being typically 47% for males and 42% for females. But these numbers are not absolute. There is also individual variation which we must account for. Plasma represented about 55% of the total blood volume. I also want to mention a term, vesicles, which is this white thin layer that separates red blood cells and plasma layers. It is made of white blood cells and platelets, and these were together less than 1% of the total blood volume. So next, I want to mention just briefly about how do we separate blood into these layers. Well, you might have guessed this method used for this is, of course, spinning the sample in the centrifuge. I also want to show this microscopy image. This is what a typical blood smear would look like. You can see the plasma in between the cells, so this white space, this is the matrix, and everything else, all other components of the blood, are dissolved in it. These include the following: a lot of red blood cells. You can see these as numerous red dots. White blood cells get stained during the microscopy slide preparation process, with their nucleus being in particular dark blue in color. Another noteworthy characteristic about white blood cells is that they are physically much bigger than the red blood cells, which we just spotted earlier. Finally, I want us to try to identify a few of the platelets. These are small cell fragments, and just like white blood cells, there are only few of these. Platelets play an important role in blood clotting. What I would like us to do next is that we should look at each of these in a little more detail. Let's start with plasma. It is a yellowish colored, slightly sticky fluid. Please note that about 90% of plasma is actually water. You might remember how we discussed about the importance and properties of water earlier in our introductory biology courses. I have added a link to a video regarding this to the end of this video in case you want to revisit this concept. There are a large number of materials that are dissolved in the water that makes up the plasma. So in the interest of time, we are not going to go through all of them, as this would be approximately 100 dissolved solutes. Instead, let's focus looking at the most important ones. I have listed proteins as the first group that I want us to look at. They are the most abundant solute in plasma and tend to remain in blood because they are not taken up by cells. Most of these proteins originate from the liver, where they are produced. A couple of important ones that I wanted to mention are albumin, which represents about 60% of all plasma proteins. These are carriers, buffers, and play a role in the regulation of osmotic pressure. Another one that I want to mention is globulin. This plays an important role in transportation and immunity. And then we have proteins that play a role in blood clotting. Then we have numerous nutrients, gases, hormones, waste products, and inorganic ions. That's all I wanted to say about plasma. Now let's talk a little more about the formed elements in blood. Most of these originate in red bone marrow as stem cells, but soon specialize to different paths. So the term that we use for this is hematopoiesis. The term poiesis refers to making of something, and the term hemato obviously refers to the blood. These cells have a rather short lifespan, just a couple of days. Therefore, there is a constant production of these going on in the body. Let's talk about red blood cells. These are known as erythrocytes, as you can see at the heading on the top. So the production of red blood cells would be therefore known as erythropoiesis. So following the same logic as before, the term poiesis standing for making of something, and the term erythro for the red blood cells. If you want to describe these cells' shape, they are described as biconcave discs. So what does that mean? Well, they are like round balloon that has been squashed in the middle, but not quite the full way through. The shape is actually important. Remember how in anatomy and physiology, form and function were always related. The shape of a red blood cell is optimized for gas transport through hemoglobin and also so that these cells can optimally squeeze through even the narrowest blood vessels. Another interesting thing to note about this is that there is no nucleus in these cells, so they are called anucleate. How can a cell come to be without a nucleus? I thought that all cells need a nucleus, right? Well, actually, red blood cells start by having one during early stages of their development, but later they lose their nucleus. Notice also that there are no cell organelles in red blood cells. For example, no mitochondria. And the reason for this is actually quite a clever one. If there is no mitochondria in a cell that transports oxygen, it will not end up consuming the oxygen that is meant to transport. Makes sense, right? Red blood cells contain hemoglobin, which you might have heard about earlier. In fact, about 97% of the red blood cells volume, not counting the water, is hemoglobin. So it must be a very important part of this. Well, I guess that the next logical question is then, what is the function of hemoglobin? It is, of course, to a large extent, the gas transport. In addition to transporting oxygen from the lungs to the body, hemoglobin also plays a role as a pH buffer and participates to the transport of carbon dioxide away from the tissues to the lungs. And I also want to mention about the word hemoglobin itself, noting that the heme part comes from the red pigment, which actually contains iron. And the globin part of the word refers to the protein that is bound to. Finally, note from the diagram that each hemoglobin can carry a maximum of 4 oxygen molecules. I want to say a few words about the color of the blood, and this has to do with the hemoglobin too, so let's have a look at that. When oxygen is loaded to the blood in the lungs, these red blood cells would now be carrying oxygen, and we talk about oxyhemoglobin. This kind of blood is brighter red in color. And then again, when oxygen has unloaded into tissues, these red blood cells no longer carry oxygen and they are called deoxyhemoglobin. These have a much darker red color, making the blood almost slightly bluish. You might have seen this difference when looking at the surface blood vessels in your own body. Now, I want to mention just a few words about why smoking is so bad for you. What actually happens can be explained by what we just learned, so let's consider that. One of the reasons why smoking is so bad for you is that cigarette smoke contains carbon monoxide. This is a colorless, odorless, and tasteless gas. It is very dense, and in case of carbon monoxide poisoning, where, for example, a fireplace has not been put out properly, it lands to the bottom of the room because of its density. In fact, out of all poisoning deaths, about 50% are due to carbon monoxide. So what happens is this: the carbon monoxide takes oxygen's place in a red blood cell's hemoglobin, blocking oxygen from binding to it. Therefore, we end up with this resulting in impaired oxygen delivery and utilization. Kind of dramatic, huh? I also want to talk a little bit about the production of red blood cells. This was known as erythropoiesis. As we saw earlier, remember poiesis for making of something and erythro for red blood cell. The production of red blood cells takes place at the red bone marrow, where these cells start initially as stem cells and then specialize further. So there can be two conditions that we must consider. If there are too few red blood cells, this leads to hypoxia, so tissues are starved from oxygen. And interestingly, if there are too many red blood cells, this results in increased blood viscosity. So what is important is that there is a balance between red blood cell production and destruction. And this depends on two factors, hormonal and dietary. Let's look at both of these in a little more detail. In case of the hormonal control of red blood cell production, one hormone that we want to look at in more detail is called erythropoietin. This is more commonly known as EPO. Erythropoietin hormone stimulates red blood cell production. It is released from the kidneys in response to low red blood cell numbers or low oxygen levels. Things that can cause these conditions would be iron deficiency, high altitude, lung problems, or amount of testosterone. Note that males have more testosterone and therefore more red blood cells. So, can we think of any event where this principle could be misused for some unfair advantage? This actually happened in my native country in early 2000s. Although high altitude training is considered as an acceptable conditioning method for a competition, the illegal use of artificial EPO hormone was widespread practice amongst cross-country skiers. And of course, the aim of this was to ensure that these athletes could carry more oxygen and so that they could perform better. This, of course, is not okay and is against the rules. It also comes at the risk of blood clotting, getting a stroke, or even a heart failure. So luckily this was caught and has now stopped, and those who needed to were penalized even retrospectively. Now let's talk about a different type of control. Control of red blood cells with diet. Amino acids, lipids, and carbohydrates are needed for red blood cell production, and of course iron too. So while free iron is toxic, it is necessary for the body. Iron is stored in our body bound to proteins. So what happens to red blood cells? Well, they live for about 100 to 120 days. And because remember, they were anucleate, this means that they did not have a nucleus. They cannot grow or divide to make new red blood cells themselves. So when red blood cell becomes too old, they are broken down in components. Some of these, such as iron, can be reused. Others, like heme, are removed, for example, by the liver. There are a couple of disorders that I want to mention before we wrap up this video. Let's have a look at those. First, I want us to discuss about anemia. In anemia, the blood has an abnormally low oxygen carrying capacity. This leads to the oxygen levels being too low for supporting normal metabolism. Typically, it is a rather sign of a problem than the sole problem itself. And therefore, the solution to these issues lies in solving the true condition behind the anemia. Some typical symptoms would include tiredness, paleness, difficulty in breathing, and feeling cold. And overall, anemias can be classified in 3 groups based on the cause behind them. These are anemia due to blood loss, anemia due to not enough red blood cells being produced, or too many red blood cells being destroyed. So let's have a look at each of these in a general level. In anemia that results from the blood loss, we can typically have 2 main types. Firstly, the ones that are due to a rapid blood loss, for example, due to a severe cut. The approach to these would be to replace the blood that has been lost. So blood transfusions are going to be important here for treatment. Secondly, the other type is a chronic loss of blood, which then results in anemia. In these cases, the loss of blood is rather a little at the time, but happens consistently over a long time. Some good examples would be hemorrhoids or a bleeding ulcer. And really, to treat these, we must address the primary problem, which then stops the blood loss and rectifies the situation. In cases where we do not have enough red blood cells being produced, there can be a few different causes. Firstly, we can have a situation where there is not enough iron in the diet, or the iron taken in is not properly absorbed. Remember, iron was needed for each of the heme groups of the hemoglobin. So as a result, we would now not produce red blood cells that would function, and they do not look like the normal ones that we are used to, but rather appear small and pale in color. And most importantly, they are unable to transport sufficiently oxygen. We would treat this situation with iron supplements. Other reason for the lack of red blood cell production could be issues with vitamin B12. So this can be either intake or absorption of vitamin B12, which then results in issues in the process where the red blood cells are supposed to divide. This would cause red blood cells that grow but fail to divide, resulting in large red blood cells known as macrocytes. So again, sufficient oxygen transport would be compromised. And this can be sometimes a problem, especially for vegetarians. And what we would do could be, for example, vitamin B12 injections or supplements. One more cause for the lack of red blood cell production that I want to mention is known as renal anemia. This is often accompanied with renal diseases where kidneys cannot produce enough EPO hormone. And the treatment would be quite simply administering synthetic EPO. Finally, there are many reasons why too many red blood cells may be getting destroyed, and while your textbook discusses quite a few of them, like aplastic anemia, where red bone marrow is destroyed or inhibited due to drugs, chemicals, radiation, or viruses, hemoglobin abnormalities due to genetic disorders, and the one that I want to focus, the sickle cell anemia. So what we are seeing here is a normal red blood cell. See the round shape, slightly squeezed from the middle, giving this biconcave disc shape that we talked about earlier. Well, in a sickle cell anemia, instead, the red blood cell would look something like this. You can see the sickle shape of the red blood cell. This is typically due to a mutation in the DNA that codes for the red blood cells. So here you can see a normal amino acid order of a red blood cell. And just a small error in the DNA, which then causes one of the bases to be coded wrong, like we see here. This would have a catastrophic effect on the resulting red blood cell shape, as we see with this sickle-shaped red blood cell image that we have here. And the problem with this is that these sickled red blood cells do a fairly poor job with carrying oxygen, and also have a tendency of getting caught and blocking some of the smaller blood vessels. So this would be a problem. Here comes an interesting point though, and this might help you with your exam question where an application of the knowledge is needed. Here is an interesting map showing how sickle cell anemia is especially common amongst the sub-Saharan African people. Why would this be important? Well, there's something else that is common in Africa. Yes, you might have guessed it, malaria parasite. See how these maps particularly almost overlap. So why would this be important? After all, sickle cell anemia was not a good thing. It made you to have a poor oxygen delivery to the tissues in your body and could cause issues with the sickled red blood cells getting caught in the small blood vessels. Well, it turns out that having sickled red blood cell may also be beneficial. The malaria parasite actually spreads through entering the red blood cell. If the red blood cell is sickled in shape, it cannot enter in it. So having this condition can be almost like a protective characteristic towards malaria. Who would have known? Well, what we normally do for sickle cell anemia is that we treat these conditions with transfusions or chemically preventing the red blood cells from sickling. So, this completes a review of the components of the blood and looking in more detail to plasma and red blood cells. In the next video, we will be looking at white blood cells and platelets. I hope to see you there.